This is Chapter 6, Part 2, How Microbes Eat, Transport Mechanisms. So, it's necessary to transport certain nutrients or the nutrients that the cell needs across the cytoplasmic membrane. This happens even in organisms with cell walls. So, we're going to kind of review a little bit on diffusion and osmosis and how this relates to this. So we know that diffusion is the phenomenon of molecular movement in which atoms or molecules, and I usually describe it as solids because it's a little bit easier to understand that way. However, you don't want to be too confused because one of the one of the examples I give is if you spray perfume in the corner of a room, that smell will fill the room eventually. And people may say, well, that's a liquid. But we're talking about the solids in it that create the scent. So diffusion is basically the movement of solids, and they move in a gradient from an area of high concentration to an area of low concentration. So when you think about that, try to picture it in your head and you say, okay, you're in the corner and you take, a perfume and you spray it 10 times, right? You take 10 pumps of the little nozzle on the perfume. And in the area that you sprayed it, it's going to be very, very concentrated, okay? But the rest of the room is going to be very, very low. There's going to be very low concentration of that perfume everywhere else in the room. And eventually, because molecules, atoms, everything moves all the time. Everything's bouncing off of each other in the air. And as that happens, they're going to get farther and farther away from each other, and they're going to fill the room until it is a happy medium. It's kind of the way I describe it, where everything is even, everything is the same concentration in the room. So it may not be quite as strong in the room 10 minutes after you spray it, but it's certainly not as strong as the area where you sprayed it initially. But that will spread out and diffuse throughout the room. All right, so osmosis is the movement of water. So diffusion is the movement of solids. Osmosis is the movement of water. And that is across a selectively or differentially permeable membrane. So I usually just describe it as a selectively permeable membrane. So it's going to be selective about what it allows through. Specifically, water is going to flow through this particular membrane we're talking about. And remember, diffusion is the movement of solids from high to low. Osmosis is the movement of water from high concentration of water to low concentrations of water. So because of this, and in the pictures coming up, this might make more sense. Because of this, I always have the little statement that says solids are thirsty. Solids always draw in water, pull in water. So if you have, one cell that you drop in a cup of water, and that cell is way more concentrated with solids than the water is. Let's say that the water is pure water and the cell is, we'll say, not 10% solids, right? So that means that it is much more concentrated than the water is. And so the water will draw into that cell going towards the solids in that cell, and then that cell will swell and burst. So, and we'll talk about this more moving forward here, but just remember that water will always move from high concentration of water to low concentration of water. So to revert back to my example with the cell has high solids, which means it has low concentration of water. A lower concentration of water than what's outside of the cell, because outside of the cell, the cup of water that you dropped the cell into is all water. So when we're talking about osmosis, the cell has passageways that allow free diffusion of water but block certain other dissolved molecules. And when the membrane is placed between solutions of different concentrations of solute or solids, and the solids or solute cannot pass through the membrane, water will diffuse at a faster rate from the side that has more water to the side that has less water. So from high concentration of water to low concentration of water. That's osmosis, the movement of water from high concentration of water to low concentration of water. And this will continue until the concentration of water is equal on both sides of the membrane. So here, inside of here, you've probably somewhere along your lifespan, you maybe have seen this experiment. Maybe you did it in school, maybe you did it, watched a video on it or something. But so you have this tube and you have this semi-permeable membrane. Dialysis tubing is something that we would use a lot in class when we did this in class years ago. And, the dialysis tubing is kind of playing the same role that the cell membrane is. So that's why it's showing in this experiment how a cell would react. So out here, you can see that these are just water molecules and nothing else. It's pure water. We don't have any sodium in there. We don't have anything else in there. It's just pure water. And in here, we'll say this solute, these little red dots, we're going to just say it's glucose, right? So let's say it's a 20% glucose solution, right? So there's all these solids in here, and they are very thirsty. Solids are thirsty. So they're going to draw in that water through that semi-permeable membrane, and then that water level in this tube will rise up because we're gaining water in this sac, and it's just going to rise up. So that's how osmosis works, where the semi-permeable membrane will allow water molecules through, but other stuff is too big, so it needs to be helped through in some way. That's the whole point of learning about osmosis in these biology classes, is to understand how giving a patient pure water versus a 50% glucose solution or sodium chloride solution versus a solution that's equal to their their consistency of their blood. So it can be detrimental if we give the wrong concentration of fluids to a patient. All right, so this talks about the isotonic, hypotonic, and hypertonic solutions and what will happen to the cell. So we're talking about microbes here. So here are the cells with the cell wall, which, this is saying without the cell wall, but same idea is going to happen. So in an isotonic solution, that means we're putting the cell into an equal concentration to what's inside of the cell wall. Okay, so it is a happy medium. We put the cell in there, water concentration is equal inside and outside of the cell. Thus, the rates of diffusion are equal in both directions. So a little bit of water enters and a little bit of water leaves. So it stays like a seesaw, really easy, really even, and healthy. In a hypotonic solution, that means we are putting pure water around that cell. Okay, so we put that cell in pure water. So the net diffusion, so if we put this cell in pure water, that means there's way more solids inside because we saw the chart or the table that showed what is inside the cell and how 97% of the solids and what they are. So there's a lot of solids in here inside of this bacteria. And so because of that, all those solids are thirsty. So water is going to move into the cell, and that cell is going to swell up and burst. Okay, and that happens in both cases here, whether it has a cell wall or not. So swells and pushes it tightly against the cell wall, and the wall prevents the cell from bursting. However, depending on if we have a cell wall or not, that's different. So, but the cell will swell up here and it's not going to be in good shape because of that. Hypertonic solution, water diffuses out of the cell. So hypertonic means we put the cell in a very concentrated solution, right? Like super salty solution. And so it's going to be more concentrated outside of the cell than inside of the cell. And solids are thirsty, so they're going to suck all the water out, essentially. The water is going to be drawn out of the cell. So water in a cell wall type scenario, water diffuses out of the cell and shrinks the cytoplasmic membrane away from the cell wall, and the process is known as plasmolysis. So that is going to be also tough for the cell to survive with that. In a regular cell without a cell wall, the water is diffusing out of the cell, causing it to shrink and become distorted. We call it crenation, like in human like blood cells or such. So you can see here the cell wall has a lot to do with helping to maintain that integrity of the outside of that cell to help it survive. But sometimes these are, sometimes they still can't survive through that. All right, so that was osmosis. So we have passive transport and active transport. So let's kind of revert back to AMP1 for a second. Simple diffusion and facilitated diffusion are types of active transport. Osmosis, are types of passive transport. Osmosis is also a type of passive transport. Active transport requires some sort of energy, some ATP is needed in the process. So, and that's mentioned here as far as active transport. And then I'm just going to try to buzz through these couple slides before I end this one. So endocytosis is eating and drinking by the cells. Endocytosis is when the cell encloses the substance in its cell membrane. So it takes it in and encloses it within its cell membrane. It simultaneously forms a vacuole and engulfs the substance. And two examples, and these should be indented here, but two examples of endocytosis are phagocytosis and pinocytosis. So again, phagocytosis and pinocytosis are examples of endocytosis. Phagocytosis is basically like amoebas and white cells. They come along and they just kind of engulf it and ingest it and later on break it down and use its parts. Pinocytosis has to do with liquids, so it's ingestion of liquids versus the solids. And I say Pinot like wine, liquids, right? If you can remember that Pinot is a wine and a wine is a liquid, then you remember that pinocytosis has to do with liquids, and then phagocytosis is more solids. All right, so we'll stop there and we'll pick up on the next part of Chapter 6.